pleasure to welcome back Jennifer Chow, CEO and Managing Director of Chimeric Therapeutics. Chimeric is an Australian biotechnology company broadly involved in the field of cell therapy targeting various cancer types. Many of you will be aware of the success of CAR T therapies in curing blood-borne cancers, including several different types of leukemia. These drugs were developed by the likes of Juno and Kite Pharma before those companies were acquired for billions of dollars. Gilead, for example, acquired Kite Pharma for US $12 billion in 2017. Chimeric is looking to repeat the success of these CAR T pioneers, firstly with an autologous T-cell therapy for the treatment of solid tumours. And earlier this year, the company released some very encouraging data from a phase 1 study in glioblastoma, which we'll hear more about shortly. The second platform is an allogeneic natural killer cell therapy targeting solid tumours and blood cancers. The portfolio includes 7 assets in development and 3 clinical trials underway. Jennifer, it's a pleasure to see you again, and thank you for joining us today.
Thank you so much, and thank you for having me at this year's healthcare conference for Bell Potter. I very much appreciate it. So I will start off with Chimeric is a clinical stage cell therapy company, much like you said, John. And for us, that really means that a lot of our assets have already been now able to move beyond the mice models, and we are in clinic. So look forward to updating you on our clinical progress. From a Chimeric point of view, our mission is all about being committed to bringing the promise of cell therapy to life for more patients. And John, I often get asked what this actually really means, or how to describe this. And I'll give you a little bit of background on what this means to us. I've worked in drug development, as has my team, for many, many years in the oncology space. And what we've seen over the course of the last, 60, 70 years in oncology drug development is a bit of an evolution of therapies. Initially, we saw chemotherapies. And chemotherapies were sort of the first approved types of therapies for oncology, but were very, very not targeted therapies that essentially really looked to just blast out and kill anything in their path. We moved in the 1990s to targeted therapies, where for the first time we were actually taking drugs and we were targeting them against specific cancers. And then in 2010, we introduced what we call immunotherapies. And for the first time, we weren't targeting cancers, we were actually targeting the host or a patient. And we were leveraging a patient's own immune system to actually fight their cancer. And as we introduced each of these different types of therapies, what we've been able to do is we've been able to improve overall survival or survival for cancer patients. When we introduced chemotherapies, we saw survival improve a little bit. And so you're really looking at the 100% of patients alive on the top left, and over time with cancer, that slowly falls. And you can see chemotherapy had a little bit of an impact. With the introduction of targeted therapies, we saw improvements in short-term survival. And then adding in the introduction of immunotherapies, we saw improvements in long-term survival. So this is really what we had seen up till about 2010. In 2017, I was very proud to be part of the group that launched the first CAR T-cell therapies. And CAR T-cell therapies really represented the first time in oncology drug development that we were targeting not just the tumor, but also the host or the immune system. So we were developing assets that were going after very specific markers on the tumor, but we were also developing assets at the same time that had the ability to stimulate a patient's immune system to fight off their own cancer. And with the introduction of these therapies, what's been so incredible is the improvement that we've seen in patient survival. And we have seen, as you see illustrated on the left with the PET scan, we've seen cancer and tumors completely melt away from patients. And on the right-hand side, you see how we've been able to improve survival, both in the short term and the long term for patients. Earlier this year, there was a publication that actually looked at patients that were treated with some of the very early leukemia CAR T-cell therapies. And very, very happily, we're able to say that those patients can actually, we can now say they've been cured. 10 years later, 10 years since they were treated, they remain cancer-free. And so this has been incredible scientific news that we have now been able to develop therapies for certain types of cancer that have been curative. Now, not surprisingly, when you see these kinds of outcomes, what you see is that cell therapy is anticipated to be the fastest growing market segment in oncology over the course of the next decade. Cell therapies in 2020 were estimated to be about $1 billion market. By 2030, we anticipate that that will be nearly a $22 billion market, and that's representing a CAGR at 31%. And you can see that in contrast to what we anticipate just generally the oncology development space to grow at. So certainly this is a fast growing market in oncology. For us, we really believe that the way to develop cell therapies is to start by bringing together experts in cell therapy development and commercialization. And so you see here the small group or the small team that we have at Chimeric. All of our team comes from the top cell therapy companies in the world: Kite, Juno, Fate, Artiva, Celgene, BMS. We have over 75 years experience working in this area across 50 different development programs. And we're all very proud of the fact that we've worked on 4 of the 6 approved CAR T cell therapies in the space. So we really have the expertise to identify assets that have the most promise and make sure they may all the way from preclinical out to the commercialization where they really can benefit patients the most. Our team spent a lot of time building a portfolio that we believe brings together the best in cell therapy innovation with broad commercial opportunity in the long run and is diversified. And you see here highlighted the portfolio. Our assets use novel designs and cutting-edge innovation in their design and invention. Our assets utilize different types of cells. So some of our assets use T cells while others use NK cells, and different types of cell sources. So we have 2 assets that are autologous, or what we call personalized, and we have other assets, 5 assets that are allogeneic, or what we call off the shelf. And so now a broad portfolio with 7 different assets in development, as John initially mentioned. This actually is one of the reasons why we're also very differentiated in the space of companies that are developing cell therapy assets, because of the diversification we have across the different cell types and the different cell sources, and certainly offers us the opportunity to have different opportunities and different shots on goal as we continue development to move forward. So I'm going to introduce you to each of the assets briefly, and also provide you a little bit of update on where we are in terms of our clinical development with each of them. I'll start with our natural killer cell therapies, and these are our NK assets. Now, these assets, as I mentioned, are allogeneic or off the shelf. So what we do with these assets is we actually start by taking cells from a healthy donor. We then isolate the natural killer cells, we engineer them, we make thousands of doses off that one healthy donor's donation, and then we freeze those cells to be able to have readily available off-the-shelf therapies. Our NK cell platform comes to us from Case Western Reserve University, and it was originally invented and designed to overcome some of the challenges that we see in the NK cell development space. You can see highlighted here some of the things that we've already been importantly able to establish with our NK cells. We use universal donor cells. We've already been able to receive approval of a master cell bank. Expansion and activation have been demonstrated, as well as large-scale manufacturing, all critical elements to be able to achieve with NK cells. We've then taken the assets into a phase 1 clinical trial, and the phase 1 clinical trial looked at patients that had both blood cancers and solid tumors. Now, this was a small phase 1 clinical trial where we enrolled 9 patients, and the primary objective of the trial was safety. First and foremost, we were very, very pleased to be able to establish that the cells were safe. We saw safety across all 3 dose levels, the patients that received cells, and no graft versus host disease. And essentially what that means is that when we give one patient another patient's cells, so here we're using a healthy donor, we want to be sure that the patient isn't going to reject the cells. So this was really important for us to establish as well. Within that trial, we were able to establish persistence of the cells and expansion, really meaning that the cells were able to stay alive and expand to be able to do their job. We saw efficacy in solid tumors, where 2 out of the 6 patients actually achieved disease control. And then notably, we saw very strong efficacy in the blood cancer patients, where all 3 blood cancer patients that were treated achieved disease control. What really caught attention here in this particular clinical trial was the response we saw of one young woman, the patient highlighted here. This young woman was in her 30s. She had acute myeloid leukemia, and she'd already received all standard therapies and unfortunately was continuing to progress in her disease. She came into the NK cell trial, had 2 infusions of the NK cells, very quickly achieved disease stability in 28 days. And by day 100, we're thrilled to be able to say that she actually had a complete response, meaning there was no sign of her cancer cells anymore. That patient went on to maintain that complete response through to 15 months. And happily, we've received an update from her center and her clinician that's treating her to know that she's actually out past 26 months and remains in complete remission with no return of the cancer. So this had us incredibly excited about the asset and as well the promise that we were seeing, particularly in blood cancers, but also in solid tumors. So what we wanted to be able to do was look for a way to actually rapidly move the development of this asset forward. We were very excited to recently be able to announce that there is a new phase 1 trial using the core NK or cells or CHM0201 in combination with an agent called vectisertib. And the belief here really is that vectisertib is going to make the tumor microenvironment more open to the NK cells and allow them to do an even better job from an efficacy perspective. So this particular trial has already been FDA approved, it's been site initiated, and we're looking forward to being able to announce the first patient dosed in the trial, hopefully very shortly. So very much a lot of excitement around the progress of this asset and this trial. Past our NK cell therapies, our first CAR T cell therapy is actually chlortoxin or CHM1101. Now, with a CAR T cell therapy that is autologous, essentially what we're doing is we're not using a healthy donor, we're actually using the patient's own cells. So here, we take the cells from the patient themselves, we isolate the T cells here, we engineer them with a chimeric antigen receptor, we freeze that patient's cells, and we send them back to that very specific patient. And so that's really how our CAR T cell therapies are manufactured and administered to patients. Chlortoxin or CHM1101 was the first CAR T cell therapy in our pipeline. This is a first-in-class CAR T cell therapy, which really means that we are the very first people to develop a chlortoxin CAR T cell therapy. The asset actually showed great preclinical promise, and so immediately went into phase 1 clinical trial. And that clinical trial is ongoing at the City of Hope Center in California. Early results in the lower doses, and so this is a 4-dose trial. In the first 2 doses, we were able to see a 70% disease control rate across the patients that were treated. So small numbers of patients, there was only 7 patients that were treated, but 5 of those 7 patients at the very low doses actually received disease or achieved disease control. And you can see highlighted here that this preliminary data for Dr. Beatty, who is the clinician that is leading this trial at City of Hope, really encourages him as we head into the higher dose levels. So here we're really working on continuing to enroll patients at the higher dose levels and looking forward to how that will, those dose levels will be complete and we'll be able to look at the data from those dose levels. The second CAR T cell therapy in our pipeline is our CDH17 CAR T cell therapy. And so this particular CAR T cell therapy, we licensed from the University of Pennsylvania last year and are incredibly excited about moving this asset into clinic. This is a novel 3rd-generation CAR T cell therapy for solid tumors, most specifically for gastrointestinal tumors, colorectal cancer, gastric cancer, and neuroendocrine tumors. Now, the team at the University of Pennsylvania have been working on this asset for over a decade, and their preclinical work on the asset was published earlier this year in the very prestigious Nature Cancer Journal as the cover story. What was shown in the preclinical models is that they were able to completely eradicate all tumor cells with no relapse of tumor across 8 different cell types. And they were able to do so with safety. So there certainly was no on-target off-tumor toxicity that was shown. So now all of the work is really focused on moving this into clinic as soon as we can to see how this plays out as we're able to treat patients with this. So with that, that really brings together what our pipeline looks like. And you can see that highlighted on the left. The green represents our NK cell assets into various stages of development. The blue is our chlorotoxin CAR T in various stages of development. And the orange, our CDH17 CAR T that is moving into clinical development. You see on the right highlighted the rapid growth we've had over the course of the last 18 to 20 months, where we've gone from having just one asset in one trial to having 7 assets in development, 3 ongoing trials with more planned for next year, 2 trials that have already shown positive early signals, and development across 10 different types of cancer. So lots of rapid growth over the course of the last 18 months. Now, looking forward, what we've also set out is milestones that we're looking to achieve over the course of the next 6 to 12 months as we go forward. Many of those milestones are clinically focused on making sure that we're advancing the clinical development of all of our assets as quickly as we possibly can to really understand the promise and the signal that they have for cancer patients out there. So for us, this is where what we look like today. We certainly believe that Chimeric Therapeutics offers opportunity, 7 novel assets in the fastest growing segment in oncology drug development. We've already seen clinical promise with 2 of those assets in phase 1 clinical trials. We have near-term value with clinical development inflection points over the next 12 to 24 months, and a team that actually has the expertise and the experience to be able to take these assets through development to commercialization and the patients that needed them the most. With that, you see highlighted on the right-hand side, our market information. We're listed ASX under CAHM. Current share price is about 8 cents, 425 million shares on issue right now, and our market cap of 35 million, with a cash position at the end of the third quarter, September 30th of this year, of $8 million. With that, I thank you so much for your time and your attention, and welcome any questions or any follow-up. Thanks, John.
Thanks, Jennifer. Great wrap there. I have a few questions for you. And if I could just direct you to backtrack a bit to slide dealing with CHM 0201, and you gave a very high-level overview of one patient there who was obviously one of the 3 patients who had some form of blood cancer or leukemia. And you described how she was still alive at 26 months and apparently doing very well by all counts. Can you tell us a little bit more about that patient? And in particular, how does this allogeneic therapy, how does this therapy differ to the CAR T therapies that are out there at the moment, for the benefit of our audience, please?
Yeah, and so John, this particular therapy, there's a couple very key differences between the NK cell that is allogeneic and our CAR T therapies. So one of probably the most obvious ways is being an allogeneic therapy, it is essentially an off-the-shelf therapy. So with CAR T cell therapies, because we're using the patient's own cells and we're sending them out to be manufactured and then returning to the patient, that whole process can take somewhere, between 3 to 6 weeks, right, to manufacture the cells. With an allogeneic therapy, we're using cells from a healthy donor. And so those cells are sitting in a freezer waiting to go for a patient. So they're much more rapidly accessible for patients, which really, for patients that have disease that is progressing, that's really important to get them their therapies as quickly as possible. So that's probably one of the biggest, from a patient perspective, one of the biggest difference between the allogeneic NK cells and our CAR T cell therapies.
Just following on from that, is there likely to be a cost? I mean, this is sort of slightly out there, but is there likely to be a benefit in terms of cost of production for your product versus the autologous product?
Yeah, absolutely. And so, I think as you can imagine, with a CAR T cell therapy, the cost to actually manufacture is quite high, because essentially what you're doing is you're manufacturing a new drug for every single solitary patient you're treating. and you have to think about the cost of being able to ship the cells to the manufacturing facility, have people that work on that particular patient cells for the course of a couple of weeks and ship the cells back. Comparatively with the allos, you're doing sort of more big scale manufacturing, and you can freeze thousands of doses at the same time. So
How
it's
can
definitely
yeah.
cost.
How can you be sure that each batch of product, though, is going to have the same level of efficacy versus the individualized product?
Yeah, and so, I think that's what all of the clinical trials are about, right, John? I think there is definitely some people that believe that allogeneic products may not have, if you went and tried to treat the exact same type of cancer that, for example, one of the approved CAR T cell therapies is currently already treating, you may not see the same level of efficacy. We actually see that there's clinical trials ongoing just in the development space to look at that. But I think that, honestly, I think what will happen is I think we're going to see a place in the market for both types of assets. There will be some cancers that will really only respond to CAR T cell therapies. And then I think there will be other cancers that potentially will respond to allogeneic NK cell therapies.
Yeah, so that was sort of touching on my next question was, isn't this problem already solved? Or are you reinventing a solution for a problem that's already done? We're already done with bloodborne cancers.
I don't think so. I think so. There are some bloodborne cancers that obviously have CAR T cell therapy approvals. And so that really is sitting inside the B cell malignancy space.
Yep.
And there is definitely, some curative outcomes in B cell malignancies. But first of all, it's not 100% of patients, right? So you're down with complete response rates somewhere in the 40 to 50% range.
Yep.
So you're obviously not, first of all, being able to cure everybody.
Yep.
And then secondly, I think that it's definitely not all blood cancers. So what we see, and for example, our NK cell trial and the patient that I was talking about is in the AML space. There are no approved CAR T cell therapies. There are no approved cell therapies in the AML space right now. So I think it's a little bit disease specific, John, really where we are from a development perspective.
Okay, so let's get out the crystal ball for a minute and talk about what's going to happen in this program over the next 12 months. How do you see it developing?
Yeah, with the NK space, with our NK trial, I think there's two different paths that we're following right now. And so the first is that we obviously have taken the NK cells and we've gone into this combination clinical trial quite quickly. Yep. And really that is because of what we were seeing in the initial trial. And so over the course of the next 12 months, we anticipate we will see the first patient, but we also anticipate that we'll see the end of that trial. That trial is planned for 12 patients to be recruited, and it should be finished by December of 23.
And
so
sorry, go on. You go on. You keep going.
No, I was going to say within the next 12 months, we should definitely have a sign of how that combination is working for patients.
And can you be very specific as to what the indication is you're going after?
Yeah, in that particular trial, it is open to patients. The trial right now is open to patients that have acute myeloid leukemia, and it's also open to patients that have colorectal cancer.
Okay, so why colorectal cancer? It's a solid cancer, I suppose, isn't it? Yeah.
It's a solid tumor. And I think there is definitely a sense that colorectal cancer patients were treated in the first trial. We saw some efficacy with them, maybe something we just wanted to enhance. And so we really believe that the potential of the combination can enhance the response that we're seeing with the colorectal cancer patients too.
And just one more question on the colorectal cancer patients. Is that you going for first-line therapy, or is this a salvage therapy? Where do you see it falling into the treatment paradigm?
Yeah, this is not first-line therapy, John. So all of the patients in this trial, whether they're colorectal or acute myeloid leukemia, would have already received first-line therapy.
Okay, let's change pace for a bit and talk about 1101. It's a 4-dose trial you've got coming up, that you've just completed or about to complete. 70% disease control rate in 5 of 7 patients, so a pretty good outcome there. And this was in glioblastoma. You had a slide in the deck there that talked about, there were some images there that talked about a patient who had apparently received drug into, it was his own control by the looks of things. One tumour received therapy and another didn't. have I got that right, or can you go back and confirm that?
Yeah, absolutely. And so the image you were looking at was from the very first dose level in the trial.
Uh-huh.
And essentially in that dose level, what we did, John, is we gave patient cells through one catheter that was placed intratumorally into a patient's brain. And what we actually saw, the image in that particular slide that you saw that where the cells were actually administered through the catheter that was placed in the patient's brain, there was disease stability. So the patient was not progressing in their disease at all. Why we actually had to say that the patient did progress was because the disease in the other part of their brain actually grew. And so what we saw is where the cells were being administered, there was no progression. Where the cells were not being administered, there definitely was progression. So that has sort of led to this hope that as we actually move on to the higher dose levels, what we've done is we've moved from giving the cells in one place in the brain to two places in the brain. And so the hope really is that by doing so, we're actually going to be able to control the disease from growing in all parts of the brain.
So there's plenty of different types of glioma. Glioblastoma is only one of them. There's plenty of childhood diseases, neuroendocrine disease. Do you see potential label expansion? You know, this is again further down the track, but is this something that's on the radar?
Absolutely. And so I think in different ways, I think into other types of brain cancer is one area to definitely explore. We also know that the cancer target that our core toxin CAR T is going after, which is MMP2, is actually expressed on other types of cancer like metastatic melanoma, sarcomas, and prostate cancer. So definitely label expansion is something that we'll be evaluating as we continue development.
So again, for 1101, what does the next 12 months hold for this program?
Yeah, for 1101, we are very anxious to finish off dose level 3 and then initiate dose level 4 and complete dose level 4. So the entire study would be completed. And then we'll be able to really assess and look at whether or not there is enough of a signal, strong enough signal to move forward into expansion cohorts, or really what the strategy becomes at that point.
Okay, so when do you see that decision being made? Is that sort of an end of calendar year 23 timeframe?
Yeah.
Okay.
I think so, John. I think, we need to finish off all of the dose level 3, 4 patients and then be able to have the data actually mature a little bit so we can look at how those patients did over a little bit of time, and then we'll be able to make those decisions.
Okay, and then one of your final slides there was in relation to milestones. It was a long list. You skipped over it very quickly. But can I ask you really just to pull out one or two key points from that slide that we can sit here in a few months' time and judge the company's performance by?
Yeah, absolutely. And so, John, I think they're all really quite clinically focused, are the major milestones. So for us, it is really having that first patient dosed in the NK combination trial is quite critical to see that trial moving on. So that would be one key milestone for us. Completion of dose level 3 and initiation of dose level 4 for chlorotoxin will be another key milestone for us. And then on the CDH17 program, it really will be about IND submission and getting FDA approval to allow us to move that program into a clinical program.
Okay, so there's plenty happening there. You've done a lot of work at City of Hope in Los Angeles. Is it fair to say that's a fairly important institution for you and continuing to conduct your trials there? What's your relationship with them like on an ongoing basis?
Yeah, I think that certainly City of Hope is where chlorotoxin was invented and founded. And so that certainly has been an important site for us. But just as much as, the University of Pennsylvania is and Case Western are as our other scientific inventing, universities or collaborations. So City of Hope certainly has led the way with the first trial. We're now working very closely with City of Hope to actually be able to expand that trial and continue to see that grow into a multi-site trial. So we partner very closely with them.
Okay. And so all that work is being done in the US now. Are you envisaging expanding beyond US borders just in the very short term?
Not in sort of the very short term, John, so not immediately in the next 2 to 3 months, but I think it is certainly within our plans. With our assets, it's always about how we expand to different geographies and when we do so. So it's in our development plans, but not immediately.
All right then, Jennifer. We are just about out of time now. I want to thank you very much for joining us again for this comprehensive rundown. It is certainly a complex story, but I think at 35 million market cap, just unbelievable value.